Welcome everyone. I thank you very much for joining us today. I'm gonna just put this up, and this is just an outline of kind of what we're gonna do today. I'm gonna start with an overview of Exchange and NMR approaches to it. This by no means is inclusive of all tools. It's gonna be a little bit of a tailored summary to guide you into the topics that we want to develop a little further, and that's going to be, I'll give you a brief introduction, and Frank Chow is really going to give you the meat of this, which is based on a set of methods and tools that he's developed while being a postdoctoral fellow in the group for the last few years. Frank has now joined another program, also part of NCI, at the Frederick National Laboratory on the RAS project. So he's moving on, but we are still doing this. And Ian, a current postdoctoral fellow in the lab, is going to actually do a practical and show you a little bit about experimental setup and processing data and fitting data. And all the software is going to be at nmrbox. That's where it is, and people that have used this before. Okay, so that's the brief outline. So the title here, I want to make certain that I sort of clarify that we're going to mainly talk in the meat of this about relaxation dispersion using adiabatic pulses. This has been developed by Frank in our group, and this treats both the NH spin system and the methyl spin system. And Ian is going to show you some methyl data. This is the more recent data that was published both last year, about a year ago, and then another paper earlier this year. And these are called GeoHard. Geo is for geometric approximation, and Frank's going to tell you about that. And hard is heteronuclear adiabatic relaxation dispersion. This is the current makeup of my group at the NCI, and you see Frank here and Ian listed here. And as I mentioned, Frank is recently moved on just in the last few months. Okay, and Frank and Ian are going to be the ones that help present today. Okay, so let's talk about timescales of dynamics and motion in molecular processes in biomolecular NMR, which is what we're focused on here and what we're going to talk about. And we all know this is from a review article from Pat Loria, and the range of motions cover an extremely wide range, from the femtosecond even of librations, picoseconds of side chain rotations, et cetera, all the way into seconds and minutes and hours in biological processes. And you can see, these librations and side chains, ligand binding, folding, unfolding, catalytic processes, and something that's near and dear to our heart, and Frank, as a graduate student in allostery, falls in this range from microseconds to milliseconds up to seconds and longer, okay? And it's this area where NMR is particularly valuable, and I think most of you that have logged into this will clearly know this. This is not new at all. And we have a range of tools. Now, in the slow end of the exchange range, the CEST, chemical exchange saturation transfer, and DEST, the dark state exchange saturation transfer, are very powerful methods. And these are in the milliseconds to seconds range. These are really the slow processes. And in the faster processes, in the microsecond to millisecond range, is really the domain of Carpenter Meiboom Gill relaxation dispersion, and also R1 row, okay? And it's this area in the purple box is what we're going to focus on today. Now, I'm going to illustrate these with particular system that we've studied in the lab in the ubiquitin pathway. It involves a multi-domain protein that has a transmembrane domain. This protein is called GP78. It has different ordered domains. And this is just a model of this system, but it includes all the domains of the protein. So it's quite complex. It's rather large for NMR. It has intrinsically disordered regions as well as ordered regions. And we take an integrated approach and apply all kinds of different tools, various NMR tools, SACs, molecular dynamics. We hope to use cryo-EM, all kinds of things to understand this. And in a complex system like this, a biological system, you're clearly going to encounter, in our case, allostery, but also conformational dynamics, exchange kinetics, et cetera. So this is what we want to look at. And in a series of papers, we've taken the classic divide and conquer approach, and we've cut out the ring domain out of this system. It gives beautiful spectra, gives nice solution structures. We've done this. We've looked at the E2 in green. There's a domain out here in magenta called Q. There's a domain called G2BR. We've looked at all of these piecewise and then put them back together in some states. But the real goal is to look at all of this together. Having done that biologically, this is in the literature. I don't want to go through it. I just want to tell you that what we've analyzed, that this forms sort of a processive machine. This GP78 will recognize the E2, capture it through an allosteric interaction that changes the affinity, triggers ubiquitination to a substrate. That's a biological effect. Interestingly, in this complex, there is a negative allostery that the ring actually weakens this G2BR and increases the off rate, allows this to dissociate, come back, free this E2 enzyme, form this initial complex, and now you can get this processive machine that builds up these chains of ubiquitin. In this case, ubiquitin, the flagstone of protein NMR, is actually the little purple dot and gets built into chains, and that leads to degradation involved in homeostasis. So we did all of this, but there's still so much we don't know. And the key point here that I want to make is these are all ground state structures. This is what we get out of an NMR, a traditional NMR, out of crystallography, even EM. We get these ground state structures that are at the bottom of the energy well. And the question is, we know from other experiments along this path as we deciphered all of this, that there's a role of conformational dynamics that actually is important in driving the machine, solving this recognition problem, this affinity problem, this release, and driving this around. So what is that role and how do we understand that? And this brings us to conformational exchange and dynamics, the subject of today's discussion. So we know that there are many thermodynamically accessible states, and they occur across many timescales. Let's take a very simple system, and we're going to say that we have two states, an A and a B state. But within this A manifold, there's many substates that interconvert on the picosecond scale or even the nanosecond scale, and that I showed you in that first slide. There's a similar case in the B state. The B state is a higher energy, so it has a lower population, but it has substates as well. Now, it may be that there's a process that recognizes the B state, and after recognition, you switch from the dark blue line to the light blue line, and you see now the A state has a higher energy, and the B state is the lower energy. But the barrier to this may be such that the interconversions between A and B are in the microsecond to millisecond range. So what does that mean? Let's look at some cartoons of structures, and let's say that A is 95% of the population, and B is 5%, and they have different shapes, okay? And they're exchanging between themselves. Now what happens if you add a ligand, okay? Well, does the ligand bind to A, or does it bind to B, or does it force A into B, etc.? So you can imagine, does it bind, shown here on the left or on the right? Well, I can tell you that it does not bind to this one. That's a small population, and the major population is B. So it actually recognizes this and binds to this, okay? So we want to understand, let me go back. We want to understand and decode this because remember, this was the ground state, and that's where we did the structure determinations. Even in the divide and conquer approach, we learned a lot about that. We did not learn so much about the excited state, but we want to know either about that state or about its interconversion. Okay, so we can take the very simple case, two states, A and B, and they exchange. We know from NMR that if it's in a fast exchange, you can titrate something and you can watch the progression. This is in an HSQC peak. It's a different molecular system, but you see the case so that you have fast exchange. You can see this, you can track this, you can plot it. There's cool software. Bruce talked about this a couple of weeks ago in his workshop, and with FX and the tools, so you can get out the KEX, et cetera, from that, and the KD. So if you look at this state, A going to B, you can write KEX as this equation, and what you want is you want to get that exchange time between A and B. You'd like to know something about the population of B, and what drives this is the difference in chemical shift between the A and B states. If you have a change in chemical shift, then you have accessible all of these parameters to understand this process. So dynamics is I think, one of the most amazing and powerful tools in NMR spectroscopy. I'm just going to give you the very quick refresher course on things that most of you know, is that if we have those two states, A and B, and let's say they're populated at 50-50, if the exchange is slow, and by slow, I mean that KEX is much less than the difference in chemical shift between A and B, shown here, then you see two lines, one for each state, and they're narrow. They have the natural line width of that state. Now, coming down from the top, if you start to increase KEX, first you see the lines broaden. They coalesce at intermediate, where KEX is approximately equal to delta omega. This can be so broad you don't see it in some cases. Small molecules, you can see this coalescence. Large molecules, often this is invisible. And then as you continue to speed up, it goes to a narrow line. And so if it's exchanging fast, even if it's 50-50, you see a peak at the center, okay, at the population weighted case. So here's my E2, which was part of that big cartoon that I showed you. And actually, we know that even in the free state of the protein, in the crystal structure, it crystallized with 3 different forms in the asymmetric unit. And so, and part of that was around this flexible loop region. Now, often in NMR, this is just an average, and in solution it is, but in the crystal, it trapped into 3 states. So what does that mean? Well, suppose that these 3 states, one of them is the important one, and it's exchanging. How do we know that? Well, if we look at, I'm going to still stay with the 2 states because it's easier, is if you have A and B, and let's say the population is now 10% in A and 90% in B, in the slow exchange, it's a population weighted spectrum. So this is 10% at A, 90% here. And then as you start to exchange, you see the same thing. At intermediate, it's broadened, but it's not coalesced beyond detection. And then as you go to faster exchange, it's in the population weighted position towards B. Now, if you have low signal to noise or high signal to noise, maybe you'll see this state in slow exchange, but in the fast exchange, you don't see it, and you don't even know. Maybe it's just, you think that's only a single state, okay? You may have other information that tells you that there are other states, and it's this invisible state that's of importance. If you want to go back and look at our papers, you'll find that actually in these conformations of this part of this molecule, there is another state, and one of those states changes the rotamer. This beige structure here, as compared to the green structure, actually becomes very amenable to forming a salt bridge and lowers the energy barrier to forming that salt bridge. And so this is what captures and starts the allosteric event. So this is a recognition event, but it's actually a very low population in the free molecule. So the way we do this, and many people, this has been pioneered by lots of people, in particular Lewis K. and Art Palmer's lab, is to decode these multiple states using NMR relaxation. Now I'm going to borrow from some slides that Tony Mittermeier developed when he was with Lewis K. to give you a phenomenological view of this. So let's say we have our 2 states, A and B, and we're going to color them red and cyan. And if you think of the frequency, this guy has a lower frequency, he's walking slowly. The red runner is actually going fast, it has a higher frequency. So we're going to say, how do we learn something about this? And particularly if one of these states is only 5%, how do we understand, how do we detect that in the NMR spectrum, quantitate it, and use that information? So it relies on a fundamental principle in magnetic resonance called the Hahn echo, whereas if you let the runner start walking in one direction, you give him a 180-degree pulse, which basically turns him around or flips him in the rotating frame, and he walks back. At an equal time afterwards, he'll be back where he started. Okay, so he forms an echo. So you can utilize this and say, what happens if now the runner changes identity? Okay, so let's say that our slow runner starts off walking in this direction. He gets flipped, but now he flips and he's now become a fast runner because he's hopped from the A to the B or B to A, and now he goes fast and he gets over here. Whereas the fast runner got all the way to this point on the track when he got flipped and switched to the other state, and now he's slow, he doesn't get back. So these guys don't actually form much of an echo, and the intensity is quite weak, okay? So now let's just use the echo, and this is what Carperzell and others have done. You now increase the flip rate. So you start them, you flip them, and you flip them again. And what you see is that they're getting closer to being at the starting point. The line's getting more homogeneous or sharper because everybody is closer to the space. You can go 3 and you can go 4. And so what you see is the faster you pulse, you don't let them get very far. So even if they switch identities from slow to fast, they're staying very close to their original or this average chemical shift. And that is actually a sharper line. Now, what Lewis took advantage of, and Art Palmer's lab has also developed many tools about this, is to look at this and look at the intensities of these resonances and or their R2 effectives, the line widths. You see broad, narrower, narrower, narrowest, as a function of the pulse spacing between these 180-degree inversion pulses, and you plot that. And what you see is these are the so-called relaxation dispersion curves, okay? And if you have a large delta omega, very significant difference in the speed of the runners, you get a big effect, and you see this. If you have a very small chemical shift difference, you see this kind of dispersion. And if there's no difference, if they're coherent, they have the same rate, then there's no change. This case is the same as the fastest case, and it's flat, okay, in this dispersion. Now, this is an important element. Even if there is exchange, but there's no difference in chemical shift, it's invisible. You can't measure it. So the ability to measure exchange is dependent on both the rate of exchange, KEX, but also delta omega, okay? And so this shows you what happens here. Okay, so now let's talk a little bit more about the CPMG. And in 2 states, I show just this R2 observed, which is proportional to this line width. And of course, that can be proportional to the intensity here. And you have a single pulse. You have a string of pulses. You see this is decaying slower and now more and more. And so you see this, and that's the same effect. So what you actually do is each one of these points is an intensity from this spectrum. So you measure this intensity. This is the inverse, R2 observed. So it's 1 over the line width here. So it's larger, smaller, smaller, and you see this decay curve, okay? There's a whole suite of very sophisticated pulse sequences. I just show one from Loria, Rance, and Palmer. And it was some of the developments from Art's lab, and it shows you how you put in a proton-nitrogen correlation, all of these nitrogen pulses. These are here to manage this relaxation. New CP is 1 over the time between these pulses, okay? So you see this. And so this is the normal experiment. The conventional approach uses something called the Carver-Richards equation to analyze this curve and extract the population of the minor state, the KEX, and the delta omega. And I think you're familiar with this. Bruce Johnson showed some very nice software to analyze this kind of data in his new package called Ring. And this has been used extensively. Lewis's lab has done a lot of things and developed experiments to not only measure the nitrogen track runners, but to look at the amide protons, the carbonyls, the C alpha, H alpha, et cetera. So across the backbone of a peptide plane here, you can get chemical, and you analyze this again for KEX and delta omega. So delta omega allows you to compute the chemical shift of the minor species, okay? And we all know now that we've gotten much better at computing structures based on chemical shifts. And Lewis was one of the first to show that he actually computed the structure of a minor state based on these chemical shifts. So this is extremely powerful information. This is just an HSQC that shows you that these shifts might be large, but they might be averaged, okay? And getting out all of these states requires fitting this data, okay? And this has been applied in Lewis's lab and Peter Wright's lab extensively, and there's beautiful work in the literature I refer you to. So if everything was sweet and honey, we wouldn't be going on any further. We'd just say, go read Lewis and Peter's papers. But there are some issues. So this is our popular E2 enzyme, and we're gonna ask about what kind of reporter residues. I'm gonna point to valine 123 here, and in this case, I'm looking at the nitrogen CPMG data, okay? And so I record this spectrum, and the protein doesn't like to be warm so much, so we do it at 15 degrees. And we see, if you want to believe, there's some exchange there, but it's pretty flat. And depending on the signal to noise, stability of samples, you might say, well, maybe there's no dynamics. And I will say that this absence of dynamics is in this class of proteins called E2s has been seen by others, in particular Ralph Boland's group did some great work and did the traditional nitrogen T1, T2 and heteronuclear NOE. They did some CPMG and they didn't find anything at 25 degrees. And they said, oh, you know, these are pretty much ground simple one state systems. But we felt that there were evidence for substates, and so we wanted to look. So what does the absence of dispersion suggest? Well, we looked at CES to see if it was really low population, slow exchange, and we didn't find anything. So it's not that. So then we started changing temperature, 10, 5 degrees, and at 1.5, you see, okay, now we have dispersion, so we have exchange. So what this says is that KEX at ambient temperature, remember the red line here is only 15 degrees, not really ambient, and not in vivo 37 degrees, is actually quite fast. It's not slow, it's not hidden, and it's quite fast. Okay. So I'm not going to go into this. This is published, but we did detailed N15 CPMG studies, combined that with molecular dynamics, and we revealed that there were state-dependent conformational exchange. What I mean by that is in that nice processive diagram I showed you of the different states which we can generate in the lab by various constructs, you can see state-dependent changes in this conformational exchange. Just going to summarize this and show you that we created an energy landscape that was a combination of CPMG and dynamics. And in the free protein, this E2, this is that flexible loop just shown as a cartoon and possible states that it can occupy. That in the free protein is primarily in this so-called open state with minor populations in a closed and a partially open. If you add this binding ligand to the backside here, it tends to drop the system into this closed conformation. The energy landscape reorients and does this. If you now add another domain, the ring, which binds on this side, it now converts to this partially open, and you get this. So we have these multi-state conversions that we're able to see through a combination of CPMG and molecular dynamics, okay? And of course, you added them both, you go straight from the upper left to the bottom, okay? So this stuff is published. You can read about that. And we can actually get some populations when we combine it with the MD. Now, I showed you the temperature dependence of this. The CPMG curves are pretty weak here, and we're only able to look at this partly divide and conquered approach to this system. So while we were successful, there were real limits here. So what's the limits? And I want to spend the last little bit of my time about that's sort of a traditional CPMG approach, and it is very powerful, but it has limits. So what if the rates of motion, I already showed you through the temperature dependence, are fast? What if they're more than, say, 2,000 or 4,000 per second? Well, this is our CPMG experiment, and you see we keep adding pulses. Well, if you notice in the literature at these kind of curves, usually these things have a maximum in the new CPMG of around 1,000 to 1,200, because you have finite pulses that you're squeezing in here. You can only get so many in there. Again, this is I didn't state, but this is a constant time experiment. So you have, say, 40 or 60 milliseconds, and you got to squeeze as many as you can, and they just run into each other, and you no longer have this approach. So what do? Well, the obvious thing is, well, suppose we just go CW, and that's an experiment called R1 row and R2 row, and you'll find in the literature some elegant work from Donghan Li and Christian Griesinger, and also Art Palmer originally, but recent work from Donghan and Christian. What you see there is, well, rather than do this, let's just put it on CW, and you'll see the R2s actually lengthen as the longer it's spin locked, and that's equivalent to never letting the track guys get far away. You just hold them right there, but they're still interconverting, and they will project that interconversion onto this relaxation behavior. Okay, so this extended the time scale significantly, but there are some difficulties. One is the instrumental limitations, the amount of RF power that you can put into your NMR probe before you create what is frustratingly known as a positive smoke test. When you burn out the capacitors in your probe by applying too much RF, then you're dead, and you're dead for other experiments. So, and recently, analyzing this data was difficult. There are some recent developments from Hans Koss, my good friend who left us recently, Mark Rance, and Art Palmer, and developments that they're currently still doing to produce approximate analytical equations that can analyze this. But these are still complicated, and the numerical analysis of this is a challenge. These guys are doing good work going forward with that, but there's still some tricks to that. There's difficulty in accessing these three parameters as you want. So this is where we wanted to look at this and talk about the case of greater than 2,000 per second. This is the R1 row and R2 experiment. So it's basically looking at relaxation along an effective field when you're applying RF along this X prime axis here. The data fitting's complicated. You can read about it from our colleagues, and the KEX is dependent on the maximum fields you can put in. So if KEX is fast, you need a strong field, okay? That's where you run into the RF limitations on the probe. So it's the spin lock is limited by the hardware. So the approach that we have developed over the last few years, driven by Frank, who you'll hear from, came from Gianluigi Veglia's lab and Mike Garwood. And an earlier paper in magnetic resonance imaging talked about using adiabatic rotation. So you have the same coordinate system for R1 row and R2 row, but now these parameters become time dependent. Okay, they're not CW here, but the B1 field becomes time dependent. This precession angle relative to here is time dependent, and you have the RF the omega RF is time dependent. Okay, so this gets complicated, but let's talk about how we're going to do this. So this was, I think, a brainchild of Mike Garwood working with Gianluigi Veglia. Veglia was Frank's PhD supervisor, and that's where Frank got into this. Okay, so what happens here? Well, this is the CPMG and the comb of pulses as I showed you, and this is actually the transverse trajectory as it's refocusing. You can see the 180 pulses as a function of their frequency, and you see the CPMG going like this, but it has a clean trajectory. You can see what's happening. Similarly, in the R1 row experiment, where you're spin locking along this effective field, you can see decays with different offsets, 500 hertz, a kilohertz, 1.5. They have clear trajectories, and this leads to ways to perhaps analyze this and extract the information. So, but I've already told you what the difficulties are. And so the idea originated from Veglia's lab, that Frank was a co-author of this, and then work that we started publishing in 2016 is to use something called heteronuclear adiabatic relaxation dispersion, or the HARD experiment. We're going to measure effectively this rotating frame, R1 row and R2 row, but instead of either clean pi pulses or CW, we're going to use a series of adiabatic pulses here that we're going to repeat. We can repeat them in some to measure a decay. And this shows you what the trajectory of the spin is for different shaped pulses. So we're going to use a different shape. We're going to measure it once with one shape, HS1, and then with 2, 4, 6, 8, etc. What you see is there's a different trajectory for each pulse in the longitudinal. This is the R1 row experiment. And so this is sampling different trajectories, different spin space, and therefore it's going to be sensitive to the exchange rate according to the shape of the pulse and the trajectory of this, okay? Now, in the R2 case, the trajectories get even more complicated, but you can see what they look like here. And so the trick is, if you believe me that this is valuable, and now you're getting more information by watching the relaxation across different trajectories, you got to figure out how to analyze this, okay? So I'm going to come back to my track analogy, and I'm going to show this as, so the traditional experiments on the bottom, the CPMG, you can have the same analogy where you have fast and slow runners and they interconvert, but now they have very bizarre and tortuous paths depending on, here I've colored the shapes black a blue, a lighter blue, and a very light blue. These are different adiabatic shapes, which means they have a different trajectory in each of these panels. The question is, how do you, these are going to carry that information about hopping between slow and fast, A and B, et cetera. But how do you understand that? Well, the trick is this path is, very defined, as I showed you. We use a specific adiabatic pulse. So we have defined this trajectory. We know what it is. The question is, how do you back extract the effect of exchange on these tortuous paths? The experiments themselves are not any more complicated than CPMGs. This is a normal kind of proton nitrogen experiment where you generate this nitrogen coherence and then you subject it to a relaxation sequence, either the R1 row or the R2 row, and then you read it back as proton. And so you get a normal proton nitrogen correlation experiment, 2D experiment for each of your relaxation conditions, and the intensity of that reflects this, okay? It's not a constant time experiment, so we actually can get improved signal to noise, and we're gonna measure rates rather than intensity, okay? The data is collected with a series of different shapes. So we run one experiment with HS1, we run another one with HS2, et cetera. Ian will show you at the end of the morning how to set those up. And the dispersion is the rate as a function of the shape. So this is the relaxation rate for pulse shape 1, shape 2, 4, 6, and 8. So you see that there's now dispersion in the relaxation rates depending on the pulse. Okay, so does it work? Well, this is a different one of the E2 protons. It doesn't have quite this flexible loop out here, but this is data that Frank did on a different E2 proton. And what you see is that this is just a cartoon to highlight where you have large rates of motion and large amplitudes of motion. I draw your attention to the KEX rates. These are 23,000 per second, radians per second in the purple, okay? And in the green even is in the 50,000 per second, okay? This is at ambient temperature, and now you're starting to see this. So now we have a way, without blowing up our probes, and we'll talk about RF field strengths in Ian's segment, but we have a way to do this without blowing up the probes. Okay, so I'm just about done. I want to show you just some general guidelines. The CPMG is a fantastic experiment. It's great, say between 200 and maybe up to 4,000 per second. This is where we were in the case that we studied. I showed you those curves. It's tough, but you can get it there. R1 row can go up to say 40,000 per second before you get positive smoke. The adiabatic R1s and R2s can, in principle, and we've seen rates as high as 100,000 per second. So now you have a broad range. You see how much further along you are. CEST is good in the slow range, 50 to 150 per second. Simulations say you can get 20 to 200. Most of the data in the literature is in this range. And geometric approximation, I haven't talked about that. Frank's going to tell you about how we do the analysis of the adiabatic experiments, but this is a critical part. This is applicable to all of these timescales, from the slow range, this part of the CEST range, all the way up to these very fast ranges. And it also gives you the ability to analyze complex topologies, such as three-side exchange, et cetera. Remember, I said the path for those track runners was tortuous, but it was well-defined. And if it's well-defined, that gives you a path to understanding what goes on. Okay, so this is the current group. Frank is here. He's going to be next, but we're going to take some questions and discussion and then a break. And Ian is going to tell us about the practical aspects, your setup experiments, et cetera. I just wanted to highlight that. This is the rest of the group from a couple of summers ago. Janusz Kościelniak, some of you may know, is our NMR engineer, and then other people that work in the lab. Okay, so I want to stop at that time. I've used all my time and a couple minutes extra, and I went through this, and I want to just open it and say, if we want to have a little bit of a discussion here, we can have a discussion. I should actually go back to the one that had times on it here. I apologize. I put that one up. Oh, it stopped sharing. My bad. I need mine. this one. Okay, so I lost it. I buggered out the times. So we'll take a little bit of discussion. I haven't been watching the partition. Yeah, okay, let's do this. I haven't been watching if there's hands up, but let's open it, whoever's monitoring it, Jeff or someone. And if there's questions or comments of your own experience about using CPMG, let's go over those, and then we'll have a little bit of break before Frank tells us about how to, the theory and the methodology of analyzing this data. Okay?
Andy, we are right on, exactly on schedule, so no.
All right.
No
I was
worries.
afraid I was going to take too much time, and I want to let people complain, argue, or share their own experience.
Okay, so if anyone has a comment or a question, either raise your hand or there's a raise hand thing in the Zoom control panel at the bottom, or you can type a message in the Zoom group chat window.
What's interesting.
I think people are.
Hey, Andy.
Rajan, how did we know that you would have a question?
How are you, Rajan? Sorry, I muted myself and then I couldn't even acknowledge you. My apologies.
Hey, I gave like 3,000 milliseconds for people to respond, and then when they jumped in. Okay, this is just a comment. Of course, it's really an awesome talk. I'm dumbfounded. I don't have a question to ask, so it is a lot to absorb. But I have only one comment to add. This is reference to Malcolm Levitt's books, Wind Dynamics. I think I remember to read there the nomenclature about the fast and slow exchange. He kind of points out probably infrequent and frequent exchange would be a better way to say that, because
yes.
the time scale, when we say fast and slow, it's often confused with the exchange rate itself. I mean, so if you really see the beautiful illustration you first showed, I think the jump between the two potential wells, how often it happens and how quick, how many times per second. So that was just like thing I want to put it out there to our NMR community. He says infrequent and frequent exchange would have been a nice nomenclature we all stuck to, but like the J coupling, we are all stuck with that kind of a nomenclature. We can't get out of it anymore. So yeah,
I agree with you, Rajan. In fact, you get into this problem of what you call the fast exchange and slow exchange, and you get inverted. You can get tight. You get wound in a knot on this when you're talking about CPMG and always that fast exchange or that slow. Because now it becomes relative to your pulsing rate.
Yeah.
And so, basically it's like the rotating frame, your reference scale changed, and it would have been better. But hindsight's 20/20. You look back and you go, now all these things we know, we should have named it this and named it that. So,
yeah.
And Malcolm's an extremely clever guy, and so he's resolved a lot of these nomenclature and units and rotation operators. He's very careful, and I respect him. So I agree with you, but I think we're stuck with what we have.
Yeah, that's right. Thank you.
Andy, Jeff here. So you focused on the extended sensitivity to time scale. What about the sensitivity to populations? Are the constraints more or less the same that you would experience with CPMG experiments? That is the limit of detection of the population of the minor state.
Yeah, I mean, so I think Frank will probably show you some simulation curves where we've looked at, because when you develop a new tool like this and how you analyze it, and this is really Frank's brainchild. He came to me with this idea and said, let's try this, and it's fantastic, I think, what it does. But when you have a new tool like that, you really want to be careful and analyze it. So we've simulated a lot of data going with a wide range or randomly selected populations, KEX and delta omega, and then submit that to the analysis tool and say, how well does it extract that? And I think the sensitivities are pretty similar. I think what Frank will show you is that you have the potential to now start expanding. I just mentioned it briefly to other exchange topologies and how you do that. Then things get complicated, and you always have to be sensitive to experimental error and how much error you have. But I think in general, in sort of traditional two-site analysis, it's equally as good to do this, and you'll get the same kind of sensitivity to populations.
Andy, we have two questions.
And of course, yeah, delta omega is really the key. If you don't have delta omega, you get nothing, no matter what the populations
Right.
are. So, you know, that's a field dependent problem, and that's and also why you usually do, and I didn't go into this at all, usually you do these experiments at 2 field strengths so that you can actually get a change in delta omega and shift and get your analysis to be better. Sorry, I'll let the other questions go.
Yeah, we've got 2 questioners have posted questions on the chat. Atul Rangadurai, do you want to unmute your microphone and you can ask the question?
Trying to figure out how to look at the questions, or he can. Here we go.
Hi, Andy. I just have one question. Could you describe one measures R2 row, especially in the presence of an adiabatic pulse? Because in the traditional Owen row experiment, you have a CW irradiation that then defines your effective field direction. So you can measure your decay along the effective field and perpendicular to it to get the Owen row and R2 row, right? Could you describe how those 2 directions are defined in the presence of an adiabatic pulse?
Well, what's defined is the initial point and the trajectory. So the initial point in the orthogonal direction is the same as in a CW experiment, and then it is the field, the B1 field is now time dependent, and so it's going to process around that. But it to a large extent, I think actually Frank has a comment about this, you come back to the same point. So it's really the trajectory, but the starting points are equivalent to the time independent, or CW R1 row and R2 row.
Okay, thanks. Yep.
And the next question is from Hamid Kushapur. Do you want to unmute your microphone and you can ask Andy directly?
He may not find the mood, so he says, can you elaborate on the purpose of having different adiabatic pulse trajectories? Well, the trick is, and I showed you just briefly, and I think Frank will expand on this, is that the dispersion, the traditional relaxation dispersion, rather than being a function of like a pulse spacing, it's actually due to the trajectory. So that what you plot out is the R1 row rate as a, or you observe, you detect it as a function of the pulse shape. So you actually need to do different pulse shapes in order to get the relaxation dispersion. And it's the behavior of the exchange between those tortuous paths that the track runners take that is encoded in the rate under each pulse shape. So as Frank will, I didn't go into this, I'm gonna let Frank do this, but you have to know that trajectory, analyze the relaxation dispersion for those shapes. So it's very tied to the adiabatic shape, and I don't want to take Frank's thunder, but what you realize is, so you have a problem of analysis, but this is in fact the problem that Frank has solved, and I'm going to let him, in the next part, he'll show you how that gets done.
Hey, Andy, so following with that comment, so qualitatively, is it correct to understand that those different adiabatic pulses you choose essentially increase the total length of the trajectory in the block sphere? Is that the idea?
No, what you do is you actually measure the rate. So where you had a loop of n, you measure it with n equals 0, 1, 2, 3, 4, okay? And you measure the rate at each of those. And then it's the analysis of that rate as a function for each shape. And that's where you get the dispersion. And so the design of the shapes is important, and the analysis is dependent on the shapes. So you have an analysis that is computed for that shape. You basically, I'm going to tell you one part of what Frank's going to go into detail on is because I showed you the trajectory in those plots, you can actually take an exchanging system and calculate the trajectory of the magnetization throughout the pulse sequence.
Okay.
And that then tells you what it will be. And you're going to use that data to analyze the relaxation dispersion.
Okay.
But Frank's going to go into detail and explain that.
Thank you.
Okay, are there any other questions? Yeah, we've got a question from Diana Capdeville.
Does she want to ask, or do we read it?
Diana,
With you
Oh, there
reading.
she is.
Either way is fine. It was just, I'm curious if you would still suggest that in any system where you don't have previous NMR information about conformational exchange, would you still go first with the CPMC, look at what the CPMC is telling you, see if you get exchange, maybe play with the temperature, and then go into these adiabatic experiments? Or are there any systems where you would go to the adiabatic, like, in my simple-minded way to approach these problems is like I would do the CPMC first, no matter what, and then start. But I don't know. Maybe have a different
You can
opinion.
definitely do that. So we've looked at cases. So in the several papers, you'll see the references in Frank's slides, and I had a couple of them. We've compared CPMG and the adiabatic to see how well first, in the range where they're both effective, you have to get the same answer, and we do. We get better errors, it turns out, in the cases we've looked at. So we can analyze and we get better errors of the fitting parameters. But
when you say better, you mean smaller?
Smaller, yeah, sorry. What I would say is that in our case, we've convinced ourselves that we can use this and cover the same region. So we would just use one and do it for across. But you could clearly, what we've shown is for both the NH and for the methyl, that you will get the same answer or the same reporting in both the adiabatic and the CPMG. So of course we're a little biased. We would say use the the adiabatic, sorry, Freudian slip there. But we find that we pick up certain things, and Frank has this example. In the adiabatic, even in the range where the CPMG is, we get a better analysis in the adiabatic experiment than the CPMG. But that region of conformational dynamics is revealed in the CPMG. It's just the quantitative nature of that is better in the adiabatic experiment.
So, like as a workflow, you would expect I would, and correct me if I'm wrong, start with a preliminary CPMG experiment. If I get something that I cannot quantitatively fit, then I would start considering these other options.
Yeah, you could do that. I think the trick there is that if it's faster than the rate you sense, you could falsely say there's nothing there. You could, by mistake, you could say there's nothing there. Often you have some indication that from other data that says, well, but I believe something's going on. I need to look a little more carefully. So you could do either.
Okay, great.
Okay.
Thank you.
Okay.
I've lost my timing, but I think we might want a bio break here.
Yes, I think if there are no other questions, we'll take about roughly 10-minute break, and we'll convene at 10 minutes after the hour. And we'll start off and allow you to do the introduction then.
Okay, great. Thanks, everybody.
Thanks, everyone.